Over the course of all of our videos and throughout the course and going back to the very beginning, we talked about the importance of DNA being essential for living things. And now at this point, we want to jump to section D and look at how is DNA actually replicated? How does that process take place? And why is it essential for living organisms? And so that's what we'll do in this video. And this is for section D1.1. DNA replication is the production of two identical strands of DNA starting from one original template. It's essential to the ability for life to be able to continue through the process of passing down genetic information from one generation to the next. And without that, life wouldn't be possible. It's required for two things, the first being reproduction. Offspring need exact copies of DNA sequences from their parents, although they'll not be exact to the offspring. Every time that cell divides in the new offspring, the DNA will be the same. And so in order for that to be able to be passed, the DNA to be able to pass from parents to offspring, it has to be replicated. And secondly, DNA is required for growth. Each cell that we have and in any organism contains a full set of DNA, the same, unless mutations take place, but for the most part identical. And before cells can divide for growth or repair, the DNA must be replicated as part of the process. And so for DNA replication, it's necessary for both reproduction and growth. In order for DNA to be replicated, it requires the separation of the double helix to be pulled apart, much like a zipper. The original strands are used as templates for the synthesis of the new strands. And those new strands are formed or made by adding complementary nucleotides. So they match the original template strand, and they're added one by one and linked together. And that site of where the copying is taking place is called the replication fork. And so the DNA gets pulled apart and there's a replication fork of where this is taking place. The product then is identical, two identical DNA strands made from those original templates. And the synthesis process is referred to or called semi-conservative because each new strand conserves or has one of the original template strands. The base sequence on the template determines the sequence on the new strand, the new strand must be complementary or matching to the other. So for example, if the template strand has an adenine, then the new strand must have a thymine because that would be then complementary. Same for guanine and cytosine. This complementary base pairing then is essential for DNA replication because it ensures a high level of accuracy due to the hydrogen bonding requirements between the different nucleotides. Adenine and thymine have 2 hydrogen bonds that form between them, and guanine and cytosine have 3 hydrogen bonds. And if you remember, going back to the beginning of the year, those two template strands, those two strands of DNA are held together by hydrogen bonds. The complementary base pairing then enables a proofreading of sorts to ensure that the replication is accurate, and it removes the possibility of mistakes or mutations. And so the enzymes that are actually conducting the replication have a proofreading mechanism, and only about 1 in 10 billion bases is incorrect, has a mutation. That's very, very small number. Humans have approximately about 6 billion base pairs per one of our diploid cells, our body cells. So on average, it's about 0.6 errors per replication of DNA in a single cell. That's not even a full mutation one. And so then this helps to ensure the genetic consistency from one generation to the next by having really, really low rates of potential mistakes. The replication process is multi-stepped and is conducted by a variety of different proteins and enzymes to be able to facilitate the process. Each strand is replicated independently of the other so that the original double helix must be separated. And how it's separated is by an enzyme called helicase. It's a ring-shaped protein that separates the template DNA by breaking those hydrogen bonds, allowing one strand to be pulled through the helicase ring, and the other to pass along the side of it. It's similar in process to opening or closing a zipper, the unwinding and unzipping of the DNA. The second type of molecule is called DNA polymerase, and we'll look at these in a little bit more detail later, but this is what does the actual assembling of the DNA using those original template strands to be able to create the new strand of DNA. It moves along the templates, adding one nucleotide at a time by bringing in new nucleotides into position where hydrogen bonds can form between the two different base pairs on the opposite sides. If those hydrogen bonds can't form because it's the incorrect number. That would mean that it's the incorrect nucleotide, and so that would be a mutation, but DNA polymerase pretty much always fixes that and catches it, and then the nucleotide breaks away if it can't match. Covalent bonds then are made to link the two bases in position. So within the new strand that's being built, the nucleotides get linked together by covalent bonds, and this occurs between the phosphate group of a free nucleotide and the sugar of the neighboring nucleotide. And then new separate polymerases are used for each different strand. Again, nucleotides are linked together by covalent bonds between the sugar of one nucleotide and the phosphate of the next. Within each nucleotide and within each strand, there's different ends, and one of those is referred to as the 3' terminus end. And this is the site that's available to link another nucleotide because it's connected at the third carbon of the sugar in the terminal nucleotide. And so as new nucleotides are being added to this growing strand, I like to think of it as new nucleotides are added at the 3' end. The 5' terminus end, the opposite, is where there's a phosphate group that's linked to the 5th carbon of the sugar. And so DNA polymerase then assembles the new strands by linking nucleotides with bases complementary to those of the template, and it always works in the 5' to 3' direction, where the 5' phosphate is linked to the 3' end of the growing strand. And again, new nucleotides are added at the 3' end of that growing strand. And DNA polymerase can only act or go in that direction. Additionally, DNA polymerase can't initiate the process. It can't just start synthesizing. It's going to need a little bit of help, and we'll look at how that takes place here momentarily. Again, nucleotides are linked together by covalent bonds between the sugar of one nucleotide and the phosphate of the next. Within each nucleotide and within each strand, there's different ends, and one of those is referred to as the 3' terminus end. And this is the site that's available to link another nucleotide because it's connected at the third carbon of the sugar in the terminal nucleotide. And so as new nucleotides are being added to this growing strand, I like to think of it as new nucleotides are added at the 3' end. The 5' terminus end, the opposite, is where there's a phosphate group that's linked to the 5th carbon of the sugar. And so DNA polymerase then assembles the new strands by linking nucleotides with bases complementary to those of the template, and it always works in the 5' to 3' direction, where the 5' phosphate is linked to the 3' end of the growing strand. And again, new nucleotides are added at the 3' end of that growing strand. And DNA polymerase can only act or go in that direction. Additionally, DNA polymerase can't initiate the process. It can't just start synthesizing. It's going to need a little bit of help, and we'll look at how that takes place here momentarily. Because DNA polymerase can only act in one direction, moving from 5' to 3', it results in anti-parallel strands that replicate in opposite directions. Helicase is moving in one direction, and one of the DNA polymerases is able to follow that. The other strand actually results in moving opposite direction of helicase because of this 5' to 3'. DNA as a double helix and as a strand is in opposite directions of the 5' and the 3'. It's anti-parallel, and so this creates during DNA replication what we call the leading strand and the lagging strand. And the leading strand is pretty straightforward. The DNA polymerase adds nucleotides moving in conjunction or towards the replication fork, and this results in a continuous replication. It just follows along helicase. Pretty straightforward. The lagging strand is more complicated because helicase is moving one direction and replication must move in the opposite direction, and so the new nucleotides are being added, moving away from the replication fork. And as that helicase moves forward, then it's exposing new template DNA to be able to replicate. And so we have, I like to think of it as kind of a leapfrog, a backwards leapfrogging process where the DNA polymerase has to double back or jump back and synthesize, and then jump back and synthesize more. And so what this does is it results in a series of chunks of DNA that have been replicated, fragments of DNA, and we refer to these as Okazaki fragments after the scientists that discovered them. And this requires a continual starting and restarting of the replication process. And so that's all on the lagging strand. It's not a continuous replication because it's moving in the opposite direction of helicase, which is breaking and unwinding and separating the template strands. Within this replication process within prokaryotes, there are a number of enzymes that are essential to carrying out the process. One of those is DNA primase, and it's a type of RNA polymerase. And what it does is it's the starting point to allow DNA polymerase to be able to function. We mentioned that DNA polymerase can't start this process all on its own. It needs some primer, and that comes from DNA primase. It's a type of RNA polymerase, and it assembles a chain of about 10 RNA nucleotides on the template strand or in conjunction with the template strand to be able to provide a binding site for DNA polymerase 3 specifically to be able to begin replication. And it's used once on the leading strand, so once it gets started, then it's done on the leading strand. On the lagging strand, because of that continual starting and stopping and restarting, it's used about every 100 to 200 nucleotides on the lagging strand. The second enzyme is then DNA polymerase, and this is what's doing the bulk of the replication. It's the primary polymerase used, and it binds to the template strand and adds new nucleotides on that 3' end of the RNA primer, and then once it gets started, then just continually adds new nucleotides on the 3' end. It's continuous on the template strand until the template strand ends or until it reaches the next RNA primer. And then, as we mentioned, it also has some proofreading to be able to correct mistakes so that mutations don't take place. Two additional enzymes include DNA polymerase 1, and this is an example of a molecule called an exonucleocase because it breaks bonds between nucleotides, so that's the exo part, and then it can also serve as a polymerase because it can link nucleotides together. And so what this molecule does is it binds the junction between the 3' end of the Okazaki fragment and the 5' primer, and it does this by removing the RNA nucleotides and replacing them with DNA nucleotides. So it's getting rid of the RNA and replacing it with DNA within those Okazaki fragments. And then lastly, DNA ligase then is what is used to be able to connect the different Okazaki fragments that are now all DNA. And it does this by making sugar phosphate bonds between the two adjacent nucleotides. And so DNA ligation is the process of joining things together. And so that's what DNA ligase is doing, is helping to stitch together those Okazaki fragments. In that proofreading process of DNA polymerase 3, if it comes across or identifies that there's an incorrect nucleotide, it moves back along the template strand by just one nucleotide, removes the incorrect one, and inserts a nucleotide with the correct base in order to facilitate the complementary matching that's required between adenine and thymine, guanine and cytosine. Over the last few decades, we've really seen the techniques and the abilities with DNA and analyzing DNA to really increase in potential. And now we're starting to see in science lots of different genetic engineering and different analysis techniques, the introduction of CRISPR, to be able to use and understand DNA at a much deeper and more advanced level. And one of the techniques that was developed in the early 1980s by a scientist named Kary Mullis was the development of PCR, or polymerase chain reaction. And this process is essential even still today to be able to study DNA because it allows for an automated way of replicating DNA but through the use of a machine, and it can produce thousands of copies of DNA very quickly and all automated. And so it's really essential for modern biotechnology. It requires only a very small amount of DNA in order to be able to work, and it has a sequence of steps that are triggered by changing the temperature in a device. It's also called a thermal cycler because the device essentially is changing temperatures very quickly in order to be able to facilitate these different stages. And so there's kind of three main stages to this process. The first is a melting or a denature stage, and this is when the temperature is usually heated to about 95 degrees Celsius for 30 to 60 seconds. It breaks the hydrogen bonds within the DNA molecule, but not the covalent bonds between the different nucleotides. A molecule called Taq polymerase, or Taq DNA polymerase, is used for PCR for replication of the DNA because it does not melt at these high temperatures. Our second stage is called the annealing stage, and this is when cooling allows the binding of primers, which is a short nucleic acid sequence that provides a starting point for DNA synthesis. It's kind of like a way to identify the specific section of DNA that's going to be replicated. And then the last stage, the third one, is called elongation, and this is the heating to 72 degrees Celsius because it provides the optimum temperature for that Taq polymerase molecule to be able to react, and then binds a single strand of DNA to the primer and starts facilitating the creation of a new DNA strand. And so these three stages are repeated over and over, and every time one of those cycles of these three stages takes place, it doubles the amount of DNA. And so very quickly, from a single piece of DNA, you can get hundreds or thousands of copies of DNA for a very specified specific region. Additionally, mixtures of DNA can be added to the machine. You don't have to have just the specific target sequence that you want to copy in the machine, because the primers are helping to identify the specific regions or sections that you want to copied. And that primer is usually about 18 to 30 nucleotides long, so it identifies a specific section of DNA to then be copied. The Taq polymerase is essential for this process. It wouldn't be possible without it because it's able to withstand those high temperatures that take place during the process. And it evolved in a bacterium that's found in hot springs of Yellowstone that has adapted to live in those high temperatures around 80 degrees Celsius. The products of then PCR can be used through a second process called gel electrophoresis to allow for the separation of the DNA molecules based off of length. And this is conducted in a gel. It's kind of like Jell-O, a little bit of a thicker Jell-O. It's usually about 3 to 4 millimeters thick, and it has holes, or wells that we call them, where the DNA sample can be actually loaded into them. And it's submerged under a solution, and it's provided some voltage to be able to create an electrical field. And that electrical field then causes the charged molecules, the DNA, to be able to move through the gel as a negative charge from the phosphates within the DNA molecule moves towards the positive end. So DNA is a little bit negative and moves towards that positive, and the application of an electrical field causes that movement. The gel within it has some filaments that resist molecule movement, and so that then allows small molecules to move a little bit faster than larger in a given set amount of time. So then this allows DNA fragments to be separated based off of their length. And after the DNA movements, the gel can be stained with dye to be able to visualize the different DNA samples within their wells, and the molecules of the same length will move the same distance, and so they form a visible band. And so after the dyeing, the staining of the gel, you can see actual different visible bands within the gel substance, and the number of bands then indicates how many different lengths of DNA were in that sample. By then comparing those different DNA samples, we can draw a number of conclusions, and we always use, or almost always use, the right-hand lane or well to be able to use a template of known distances so that we have something to compare to, and we call this the ladder. And so we can compare our unknown samples, or the ones that we're testing, to this ladder to be able to have an estimation of the size of the DNA molecules. So the use of gel electrophoresis has some big advantages, some uses that we can do. And one example would be testing for coronavirus. Maybe you had this happen in the past. A swab is used to take a sample from the nose or the throat, and then that swab is rinsed in a saline solution to be able to produce a liquid sample. The RNA in the sample is then converted into DNA using reverse transcriptase. PCR is then used to be able to amplify the sequence, so you get lots of different copies, and there are markers for the particular strain of coronavirus. It requires about 35 cycles within that PCR machine, so it produces a large number of copies. And then fluorescent markers are attached to the DNA and are observed, and if that fluorescent level rises above a specific target level, then it indicates a positive test. Advantages of using this is you can have very small quantities. If the very small quantities of the virus present, a viral DNA, will yield positive results, so it's very sensitive. And it can also be very specific in that you can use different or specific primers to detect one specific strain. Disadvantages would be that it requires materials and equipment that are expensive and have to be in a lab typically, and it takes some time. It's not an immediate test, so that'd be a disadvantage. A second example would be in paternity testing, and this would allow for the distinguishing between individuals by using sequences called short tandem repeats. And these are sequences usually between 2 and 7 bases that are repeated consecutively and that vary between individuals. So from one person to the next, how many times those are repeated is going to be different. For example, on chromosome 5, there's a sequence of TAGA that's repeated about between 6 and 15 times, depending on the person. There's usually about 13 tandem repeats that are used in DNA profiling, and it's extremely unlikely that 2 humans would have the same number of repeats. So we can use that to be able to distinguish between one person and the next. The stages that are needed to be able to produce a profile: 1, you have to collect the sample from a known or an unknown individual. That sample has to be amplified by PCR, so we have a large number of copies of those short tandem repeats. That product then is separated using gel electrophoresis by the length of the fragments, the patterns of DNA. And then those patterns, because they'll always be the same for each individual, based on those tandem repeats within the DNA of the individual, we can use that to be able to draw some conclusions. And so then for paternity testing, the bands in a child's profile must match that of the mother and then whoever is the father. And so we can detect the individual that's the father of the child based off of the bands, because the child is going to have the same makeup of bands from mother and father. Those combined to produce the child, and a different individual that has different series of bands would indicate that it's not the father, because the child is getting its DNA, including the short tandem repeats, from mother and father. So the replication of DNA is essential to life, and in our next video we'll take a look at how that can be manipulated or changed a little bit, and also produce variation within individuals within a species.